Welcome back, friends. In the previous video discussion, we have talked about the self-splicing event and the mechanism of self-splicing. Now, in this particular video, we'll be talking about uh, the spliceosome mediated splicing event. Now, this spliceosome mediated splicing event is based on the process that an mRNA is there, which is called the pre-mRNA. Now, this pre-mRNA will bind with some of the proteins. So not only proteins but the protein nucleotide complexes. They are also called ribonucleoproteins or RNPs. And inside the nucleus there are short segment of this ribonucleoprotein along with the segments of ribonucleoproteins. It is called the short nuclear ribonucleoproteins or snRNPs. Okay, so let me write it. They are called snRNP or short nuclear ribonucleoproteins. So these short nuclear ribonucleoproteins or snRNPs are very important protein molecules or very important mediators for this spliceosome mediated cell splicing. Now for any kind of spliceosome mediated splicing, we must have the complex formation between this snRNP proteins and the pre-mRNA. Now the pre-mRNA that we are having, again it is consisting of introns and exons. Now we are going to see the mechanism in detail, and this mechanism will be seeing that the proteins like snRNPs will allow to recombine, will allow to hold on to the 3 points, 5' splicing point, 3' splicing point, as well as the branching point to bring them closer. Now this close proximity of these 3 branch points, 3 points, one is the branch point and one is 5' splicing point, another one is the 3' splicing point, in turn help to catalyze the reaction faster. Now remember one thing, in any kind of splicing, whether it is a self-splicing or it is a spliceosome mediated splicing, no protein molecules like these snRNPs or whatever the protein mediators are, they won't act as a catalyst. No protein molecule act as catalyst in the splicing reactions. In turn, they can only give the structural rigidity to the system. They can only provide the structural role for the splicing event. For example, to hold the different branch points together, to hold the reaction centers in close proximity, and rest of the part, the catalytic part, is only present in these intron sequences. So whether it is a self-splicing or spliceosome mediated splicing, the actual catalysis is performed by the sequence specificity that is present in between the introns. So this is a very important concept. Rest of the part, in case of spliceosome, it is forming a complex structure along with this snRNPs and other factors, another cofactors, and another different types of protein or non-protein factors along with this pre-mRNA to form a bulge complex. This bulge complex is called spliceosome. Okay, now in electron microscopy you can see the spliceosome something like that. So say you can find so this is the mRNA, you can find structure like this. So this kind of loop like structure you can see in electron microscopy, it is telling. And also not only loop but also you can find small molecules for producing bead like structures. Now these bead like structures to form this loop coil is the denotation that splicing is going on. So this bead like structure are simply this snRNP and RNA complexes that are forming during the splicing events. So in this kind of splicing, again we need proteins like snRNPs. The different types of snRNPs are EU or other proteins like recognition factors. Enhancer region is there, so there are also proteins like SR proteins, something like that. So there are different varieties of proteins and factors are required for the completion of this process by the by this spliceosome mediated system. Now let us begin with this drawing. So I have drawn the whole picture about this spliceosome mediated process. Now let's begin from the first step. So this is the step number 1. So the step number 1 is this, and the very first step we just draw the pre-mRNA. So it is having 2 exons and 1 introns. So this red color region is called introns. Okay, so 2 exons and in between we are having introns. Now it's similar like the self-splicing event. Both in between the introns and exon join place, we are having the cleaving signal or the splicing signal, which is between two G's, A G G, right, this sequence AGGU. So in between two G's, we are having this splicing signals. So here this dot represents the 5' splicing signal, and this dot represents the 3' splicing signal. Now in between the introns we are having what is called the branch point. Again, in this case, the branch point, there is a residue, specific residue is adenine. Okay, now the hydroxyl of the adenine can initiate the reaction. So let me draw it. The hydroxyl of the adenine, again here also, can initiate this nucleophilic attack, and it will help to produce a lariat structure, right? Now again, in this case, we are going to see the lariat structure. So this is the basic design of the pre-mRNA. Now the first step of this reaction is attachment or the involvement of the proteins U1 and Bbp. Now, U1 is a type of snRNP or short nuclear ribonucleoprotein. So here let us draw it. So U1 come and bind, as well as Bbp, which is also called the branch point binding protein. So branch point binding protein Bbp along with the short nuclear ribonucleoprotein type U1 come and bind with two regions. Now Bbp, as the name suggests, it will bind with the branch point. On the other hand, U1 recognizes the 5' splicing site and binds with that. Okay. Then what will happen? Another kind of this snRNP, which is U2, comes in, and it will bind to this branch point, and it will kick Bbp out from the branch point. Okay, so this is the second step. U2 comes in, it will kick the Bbp out. So now what we are having a pre-mRNA complex. In this pre-mRNA, we are having two exons at two ends. In the between, we are having the intron region, and in these intron regions, we are having two ribonucleoproteins bound with it, U1 and U2. U1 is attached to the 5' splicing site, and U2 bound with this branch point. Okay, so this is the situation. After this complex formation, after the formation of this first spliceosome, which is via the intron exon as well as with the short nuclear ribonucleoproteins, in this situation, this whole system encounters a tri-snRNP complex. Tri-snRNP complex made up with U4, U5 and U6. So these three ribonucleoproteins come closer. They form a tri, or they form a complex, a complex like structure. Now this tri ribonucleoproteins U4, U5 and U6, they will come and they will bind with this intron exon interacting region. As they start to bind, they start to displace some of the previously bound ribonucleoproteins, because these ribonucleoproteins are having much more affinity towards this RNA. Now why we at all require this ribonucleoprotein instead of simple proteins? Now when you're talking about ribonucleoprotein, we are talking about proteins which are having RNA segment on it. So we are talking about this type of mixed molecules. So proteins with RNA regions on it. So it's a protein and RNA complex. That's why we call them ribonucleoprotein. We require this kind of complex protein structures because we are dealing with RNAs, and we know that RNAs are single-stranded. So most of the time RNA can bind and form bonds with hydrogen bonds with itself, because there should be complementary bases somewhere, so they can intrafold between itself. Okay, self-folding of RNA can be seen, and as a result it can produce some so the RNA may not be accessible that much. So if you utilize these proteins, this RNA backbone of this protein will help this protein to zip up with this pre-mRNA. which is to be edited. That's why we require ribonucleoproteins instead of simple proteins. So now let's come back. So it's a tri-ribonucleoprotein complex. It will come and bind, replace the U1, and U4 is also released. Now U4 is something which you must understand this is the mechanism you understand. So what will happen, as it is coming, U6 is going to replace U1. Clear? U6 is going to replace U1, but U4 and U5 is there. U5 simply bound with U6, but on the other hand U4 is also released. Now here comes the important part. Here you can see after this step. So after this step, what we can see here, U6 takes the part of U1, U5 is simply bound with U6, but U4 is dissociated and U2 is still there at the branch point. So what is the need of carrying a protein which is not required, right? So by looking at this picture, what we can get, what idea we can get, that U5 and 6 is there, which is required, fine, we bring them. But what is the requirement for U4? That simply we bring them and then just kick it off. The answer is yes, there is an importance. Remember, all of these ribonucleoproteins are having a RNA chain, so it can bind with RNA. It's fine. So as they are having RNA chain, two ribonucleoproteins also can bind. So let us consider it like this way. So this is another ribonucleoprotein. This is, say, another chain. So these two ribonucleoproteins can bind with each other with the help of their RNA complementary nature of their RNAs. If they find any kind of similarity between sequences or complementary sequences, they can bind, right? So here, whenever we're talking about forming the tri-ribonucleoprotein complexes, that means these protein complexes are bound with each other via this RNA region. So they are having complementary in the RNA region nature. So if we think these are the protein complexes, they already zipped with each other. Now U6 is not, remember this, U6 is not accessible to the region where U1 is residing, because its RNA region is already occupied by U4. So let us consider this is U6 and this is U4. So this U6 and U4 are attached with each other via the RNA linkage. So as this is happening, this U6 is not available to bind with the RNA because its RNA region is already occupied. So what we need to do, we need to cut this U4 out so that this U6 can bind with this pre-RNA. This is what is happening here. U4 is cutting out, so now U6 can bind with this pre-RNA via the RNA complementarity, and this binding kick U1 out and U6 just placed in the place of U1. Okay, understand it? Right. Then this U6 is also, now as a result of this U4 leaving the U6, the whole RNA region of the U6 is free. So U6 can bind two regions. It can bind with pre-mRNA. Second thing, it can bind with U2. So now instead of binding with one U4, it can bind with pre-mRNA, along with it can bind with U2. So this triple interaction of U6 is a very, very crucial for this kind of mechanism, because this is the step which allows three important points to come close to each other. Because if these 3 points are not coming close to each other, the reaction will not happen. Because remember, in any catalytic reactions we need close proximity, longer time to be happen. Now in this case, by removing U4, we are opening the RNA chain. So this RNA chain can in a same time attach with pre-mRNA, kicking U1 out, as well as it can attach with U2. Now it place itself. This placing of U6 helps to bend helps to bend this mRNA, pre-mRNA, to produce the lariat structure. Remember, now this bending will allow to take these three segments close to each other. Remember, what are the segments? So this is the 5' excluding signal. So this is the first, this is the second 3' splicing signal, and this is the branch point. So you can see in this case they are far apart. So in case this is first signal, this is second signal, this is third signal. But here, whenever we are folding it, it actually comes really close to each other. So for bringing them closer, we need U1, because U1 is a pretty large molecule. It can bind with mRNA as well as U2. That's why we need to open U1, U6. So there is a control, because if all the time U6 is opened, they are going to bind with any kind of molecules and then bring them together. Splicing will happen. So we need to control splicing, right? So if splicing is happening all the time inside the cell, simply it's a wastage of energy. So we need to conserve this. For that, we need to block U6 with another snRNP, and that supporting role is played by U4 in this case. Okay. So now, after attaching with U6, U5, this complex is formed. There's a loop is formed. After the formation of this loop, this A, with the help of the 2' hydroxyl, so let me take another color, with the help of 2' hydroxyl here, this 2' hydroxyl of is adenine can act as nucleophile, so it will attack this region, it will attack U1 region, and as a result U1 will be cleaved. So the E1 part is cleaved, or the exon 1 is cleaved, as well as this terminal region is attached to this region to form a lariat structure. Get it or not? Simple. Just think about the process of self-splicing. Now rest of the part is simply resembling the self-splicing process. Now this part is the important. This part is in the unique in this case. So the trinuclear snRNP complex formation is a unique part about this spliceosome mediated process. But rest of the part is similar to the self-splicing. So again, this 2' hydroxyl will attack this to form the lariat, as well as the E1 will be cleaved. Now if we cleave E1 here, the E1 can be dissociated, the E1 can be spread, the E1 can diffuse through the one place to another place inside the cell. So we not only can cleave it, we must have hold E1. So for holding this E1 segment, we need U5. So this is the job of U5, to hold E1 when it is get cleaved. So we cleave this E1 using the 2' hydroxyl of the branch point. E1 is cleaved, or exon 1 is cleaved. Now U5 hold on to this E1. Now remember, after the cleavage of E1, we are having two parts. This 5' region of E1, it consists of phosphate. On the other hand, after the cleavage from this side, it generates a 3' hydroxyl also, right? Now this 3' hydroxyl can act as another nucleophile, as the second stage of the reaction. So it's a stage 2. This is stage 1, this is stage 2. In this stage 2, this hydroxyl will attack this place, a 3' splicing site. Okay, so here now this hydroxyl, 3' hydroxyl of exon 1 will attack the 3' splicing site. Then what will happen? It will bind with E2. So now E1 binds with E2, and then lariat structure is removed. Now this lariat structure which is removed here is also bound with 3 proteins. U6 previously bound, U2, U2-U6 complex, and U5. So ultimately what we can get is the exon 1 exon 2 complex, which is our desired product, which we can also call as mature, remember, mature mRNA, which can code for proteins via the translation, and then lariat structure along with the snRNPs. Now again in this case also this products will be removed. Exon 1 exon 2 will be removed from nucleus to cytoplasm for the translation, so product removal. And laddiates can also be degraded to form SNRNPs or snow proteins, okay, not snow proteins, snow RNAs, sorry for that. And this other protein complexes, SNRNPs, or in short form they are also called SNORPs. They are also called SNORPs. The SNORPs are removed and they can be utilized in the future processes. Now this is a basic process of spliceosome mediated splicing, but there are also detailed steps. Detailed steps, for example, there are sequences that are present in these intron regions which are responsible for the binding of some protein factors. Now these protein factors binding with those segments or those sequences enhances the splicing event. We call those signals as exonic splicing enhancers. Okay, so there are enhancers present there, which enhancer sequences are present where some proteins can bind, and binding of some proteins to this enhancer will enhance this process. There are also some inhibitors sequences that are present. So remember, this enhancer or inhibitor, whatever we are talking about, they are simple sequences and they are present in the introns or exons. Most of the time they are present in introns, so they are not proteins, they are sequences, nucleotide sequences. Proteins can bind with themselves, so when protein bind with those, it can trigger, it would be, the effect would be positive or it can be negative. Okay, so if an enhancer binds, the effect is positive, it is enhanced. If an inhibitor binds, the effect would be negative or it will be inhibited. So this is a process and this is a mechanism of spliceosome mediated splicing event, and I hope it will help you. Thank you.